Well, Biology 30s, here we are today, and we are going to be talking about a dihybrid cross. So in our last genetics videos, we learned about a monohybrid cross where we were looking at one trait. So this stuff is in your textbook, page 591 to 593. It's a good idea to give that a read. And then understand that when I say a dihybrid cross, the word di, that prefix, it means two. We're looking at two different traits caused by two different genes within a single cross. So the good news is you already know how to do monohybrids, so you also know how to do dihybrids. It's not going to be very much different. It's kind of like two monohybrid crosses, and so I'll show you a shortcut method for that later. Okay, a couple of important things is the rules for doing these questions. Always make sure you're using the same letter for a given gene. You're sorting the alleles into gametes properly. So here, this word from math, foil, is going to come into play, and I'll be talking about that. And another note, you don't have to repeat similar gametes when you're doing your Punnett square. Okay, so that'll all make sense in just a second here. Right now, we're going to begin with an example problem. So to start, we have a homozygous tall and homozygous green pea plant, and we're crossing it with a pure breeding. What does that mean? It means purebred. It's homozygous, both for short and also for yellow. And now here it tells you that tall and green are dominant. So we can set up our traits, right? We have the height of the pea plant, big T for tall, little t for short. The dominant, so the tall is dominant too. That's what that greater than sign means, the short. And then we have the color of the seed. That's our second trait. So two traits here, not just one. We have big G for green, which is dominant to the little g for yellow. So as a reminder, our very first rule said use the same letter. So we've got big T for tall, S for short. No, we don't do that, right? We use a little t for short. Same letter, just lowercase. So that's why it's big G for green and a little g for yellow. We're not using a little y for yellow. Okay, so now we have our parents set up. And so from there, we're going to write down the genotypes of our parents. We know what their phenotypes, their appearances are, tall or short, green or yellow. But now we have to write the letters, how the alleles are paired up. So here we have this homozygous tall, that's big T, and homozygous green, that's big G. So that's the double homozygous dominant. And we're mating it with the homozygous short, little t, and homozygous yellow, little g. Then we have to do the gametes. Well, what are we having for gametes here? Well, we only have big T's and big G's. All four of the gametes produced by this parent is big T's and big G's. And what about the short and yellow parent? Well, little t's and little g's, that's it. So here's where rule number 2 comes into play. If you want, you can do a big 4x4 Punnett square and put here's parents' gametes from the whatever female parent, big T, big G, big T, big G, big T, big G, all the way across. And then you can take the other parent, little t, little g, and put it all the way down the side. Why bother? What's the point of doing that? You can clearly see that every single offspring here is big T, big G, little g. 100%, right? All 16 out of 16. So why would you bother doing that? Why would you not instead just go like this? Here's big T, big G, and here's little t, little g. Just do a 1 by 1. Now remember, you're keeping the t's together, the trait for height, right? Big T and then little t. And you're keeping the g's for color together, right? Big G, little g. What do you have? They're all tall and they're all green. Does not need to be done in a 4x4 when you can easily do 1 by 1. Okay, and so that's what we mean. You don't have to repeat similar gametes. Why bother doing a big giant Punnett square like that when you can easily just do a 1 by 1 and save yourself the time? The results are the same. Okay, so reminder again, you always use your capital letters first in each pair. So if we're looking at tall versus short, big T and followed by little t, we would not write little t, big T. That is not correct, right? We are going to go capital letters first and then small letters, capital letters and small letters. Okay, so always the dominant, the capital, uppercase, and then the lowercase for each pair of alleles. Okay, so let's take a look now that we have these 100% of our F1 generation was heterozygous. How do we figure out? Well, we're going to do what I call FOIL, and we're going to determine each of the gametes. So here I'm going to use some different colors. And for those of you who are familiar with math, the first letter of each pairing, F, is the first letter. So here I go. Big T and a big G. Okay, so that's how I got this first gamete. Big T, big G. Those are the first letters in each pair. And then I am going to go with the outside letters of each pair. So that is the big T, and then this outside guy here, that is the little g. So that's how I got the second gamete there. Okay, next up, I am going to go with I, inside letters of each pair. I'm doing that in orange. So the inside letter was the little t and the big G. So that's how I got that gamete there. And then I'm going to do the last letter of each pair, and I'm going to do those in black. And so that's going to be the last letter, the little t, and the last letter there, the little g. So little t, little g. So I have 4 different gametes from this parent, 4 different gametes here. There's no repeats though. It's not like we have big T, and another big T, big T. They're all 4 different separate ones. So you're getting a giganto 4x4 Punnett square. And yeah, you've got to put them all in. All of them are going into this Punnett square. And once you do that, this example is already done for you. You are then going to count up phenotypes. You can see that this entire row going down here is got a big T, so it has to be tall. Yep, big T. Each and every one of these offspring are tall. And there's a big G, so each and every one of them is going to be green. Same for going across in this column. Big T for tall, big G for green. So we've got 1, 2, 3, 4, 5, 6, 7 so far that are tall and green. And there's 2 more that are tall and green. So my total number of tall and green are 3. Next, I am going to look and see, well, now I've got a big T for tall, but a little g there for yellow. So you can see that if I have 2 little g's together, that's how I'm getting a yellow pea plant. And you can count up and you can see that there are 3 that are tall and yellow. And next up, we have 2 little t's each time there is a short pea plant. And so you can see there are 3 examples of short and green. And then who's the last guy? Well, that's the double recessive, right? And this double recessive guy, little t, little g, only one of those. Okay, so now if you're going to go and count up the numbers, let's see how many of each we had. Well, we got what is called a 9:3 to 3:1 phenotypic ratio. And this is huge. This is the Mendelian ratio, 9:3 to 3:1, right? And this is going to happen every time you're doing a dihybrid and you're crossing 2 heteros, unless there's a gene interaction. We're not learning about this this year, but when there's 2 heterozygous being crossed, you're always going to get a 4x4 Punnett square. You've got 16 offspring. 9 out of 16 are the double dominant trait. 3 out of the 16 is going to be dominant for one trait, recessive for the other, and you get this 9:3 to 3:1 ratio. So what do we mean 9:3 to 3:1? Well, the largest group, the most numbers, have both of the dominant alleles, and so they have the dominant trait, right? Tall and green in this example. And then 3 of them are going to be tall. That's a dominant trait. And then little g is our yellow. And so that is the recessive. And then 3 little t, well, that is going to be short rather than tall, and big G, so it means they're green. So this little blank here, it could be a big G or it could be a little g, okay? But as long as you have that one big G, you know that it's going to have the dominant trait. And then here's our double recessive. The 1 out of 16 is little t, little g, little g. That guy is our recessive. Okay, so what about if I'm not doing hetero times a hetero? Well, I'm going to have another video with an example like that, and I'm going to show you a shortcut method to do. And then I will have one more video, hopefully only one more, where I will have an example that I'm going to work out for you. Okay, so next video, tune in for dihybrid, where there's not two heterozygous that are mating together. Okay, till then, talk later.